It's my pleasure to welcome you back to the second annual Koch Institute Cell Signaling Technology Symposia on Signaling in Cancer. Welcome to the third session. This session's focus is signaling, cell cycle control, and metabolism, and it's my great pleasure to introduce my friend, colleague, and collaborator, Professor Yossi Shiloh. Professor Shiloh is a professor of human genetics at the Sackler School of Medicine at Tel Aviv University, where he directs the David and Inez Meyer Laboratory.
Thank you very much, Mike, and I'd like to, of course, to thank the organizers for inviting me to speak in this fantastic meeting. And I think that I will start right away by telling you the bottom line of my talk, and that is that our view of ATM is changing. ATM's claim to fame is, of course, its role in the activation of the cellular response to double-strand breaks in the DNA, probably the prototypic DNA damage response network. But in fact, new functions of ATM are emerging in various aspects of cellular metabolism. In fact, just a few weeks ago when I was writing the introduction to a paper or a grant, and these introductions tend to be generic when you write again and again on the same subject. But then I found myself writing something that I had never written before, which is ATM is a homeostatic versatile protein kinase that is vigorously activated following the induction of double-strand breaks. This is, instead of the usual, ATM is the chief activator of the cellular response to double-strand breaks. And you can also see from the title of my talk that actually doesn't contain the words double-strand breaks, but rather a more vague term, genotoxic stress, and I hope this will be clear later. But first thing first, and first in our lab is the disease that is caused by lack of ATM, ataxia-telangiectasia, a prototypic genomic instability syndrome, and I guess most of you are familiar with this phenotype. It has this striking cerebellar atrophy seen here in this MRI image, and the telangiectasias are these dilated blood vessels in the eyes, but this is in fact, as you can see, a multisystem disease, a very pleiotropic disease, and it is one of the genomic instability syndromes. In the genomic instability is best seen in peripheral lymphocytes from these patients, where you see these clonal translocations emerging, usually involving the sites of the antigen receptor genes, and this is just the early sign of the emerging malignancy, and most of the malignancies are lymphoreticular. In the older patients, we see also epithelial malignancies, and the acute sensitivity to ionizing radiation, which was first identified when 80 patients who were irradiated for treatment of cancer developed very severe radiation reaction. In fact, some of them died because of the treatment. And this is clearly reflected in these survival curves of wild type and AT cells. This is a very old drawing, as you can see, just from the fact that the X-ray dose is measured in rats. This is from my Ph.D. thesis, but this is still valid, so I guess I'll continue to show this figure. And this combination in this phenotype of one disease of genomic instability, cancer predisposition, and acute sensitivity to DNA damaging agents, all of which boil down to a defect in the DNA damage response, tell you in a nutshell the well-known connection, the intimate connection between maintenance of genomic stability and cancer predisposition and cancer development. So early on, when I was a grad student and I started to work on AT, we knew already that the radiosensitivity is the key to understanding the molecular pathology of this disease. And after a few years of work in our lab and other labs, brought us to the conclusion that the critical DNA lesion was the double-strand break in the DNA, most critical DNA lesion. So this is a very old slide, and this is what it used to look like before I was going to the next slide. But in fact, there is a new addition. And the new addition actually is because of going back to the old, very old data that I and other people had from that time, basically showing when we were screening various DNA damaging agents, basically looking for the class of damaging agents to which AT cells will be most hypersensitive. And of course, all of them turn out to be those that induce double-strand breaks. That was the common denominator. But in the course of these experiments, we tested other DNA-damaging agents, and AT cells were moderately or even slightly hypersensitive to these agents. But this moderate hypersensitivity was probably overshadowed by the striking hypersensitivity to ionizing radiation and radiomimetic chemicals. And so we sort of abandoned those data, but now we're going back to those data. They are important and they mean a lot to us, and this will become clearer in the course of my talk. And here's another point, a very important point that we have to bear in mind. Those of you in the DNA damage response community usually irradiate cells or treat them with chemical DNA damaging agents. This is very far from what's going on actually in our bodies, in every cell, in every tissue. And there are different statistics about that, but the common denominator is that there are actually tens of thousands of DNA lesions induced on a daily basis in most of the cells of our bodies. So obviously, the maintenance of genomic stability is important and is critical, but it's not exactly the kind of damage that we induce in the lab. Actually, most of it is single-strand breaks, and the double-strand break, on which we and many other labs are focusing, are at the bottom of the list with very, very modest proportion, but of course, very, very critical effect, because one unrepaired double-strand break can kill cell or lead to chromosomal translocation and be carcinogenic. So there are very sophisticated repair mechanisms, but in addition to the repair mechanisms, double-strand breaks are probably the prototype DNA lesion that activates a DNA damage response, which is emerging to be probably one of the most extensive, widespan signaling networks in the cell, in the face of any stimulus. And ATMs, of course, in the center. Just a quick reminder about the actual repair of these breaks. It is carried out either by direct joining of, non-homologous end joining, direct joining of the ends at the cost of creating microdeletions at the break site, or via the error-free mechanism, which is based on homologous recombination between the damaged chromatid and the sister chromatid, and this obviously happens in late S or G2. And there are several variations on these themes, and I will get back to them later. In addition, one major response is, of course, the activation of the specific damage-induced cell cycle checkpoints, which basically arrest the cell cycle at least for a couple of hours, wherever it is. Now, in addition to the very sophisticated mechanisms that actually activate these cell cycle checkpoints, and all of them are indeed known to be at the protein level, we recently found that actually there is a very important component which is at the gene expression level. I'll get back to that later. But the point I want to make here, and I guess you're all familiar with the basic concept of the damage-induced cell cycle checkpoints, is that in addition to these mechanisms that activate these checkpoints and arrest the cell cycle, what we're actually doing here is bringing the cell in an unscheduled, unplanned manner to almost a G0-like situation, at least for a couple of hours, which means that we have to modify cellular metabolism if we want this cell to get back from this process alive and healthy. And so indeed, the DNA damage response has very intimate relationships with cellular metabolism, and that's something else that I will elaborate on later on. And then this explains, at least in part, why almost every aspect of cellular metabolism is modulated one way or another following the induction of double-strand breaks. So this is a very sophisticated system that acts within seconds following the induction of a double-strand break in a very concerted manner. And as you know, it has several stages. And the first is actually carried out by this very heterogeneous group of proteins dubbed sensors or mediators. And I think that the only common denominator of all these proteins with regard to the DNA damage response is that they're all recruited physically to the damaged sites in chromatin, and you can see some of the activity that is going on. I'd like to make two comments about this stage of the DNA damage response. When we started working in this area, we thought that there will be maybe 5 or 10 proteins at this early stage of the DNA damage response. And we keep identifying, and other labs keep doing this more and more, to the extent that the recruitment of a protein to the damage site has become a criterion or a readout in screening for new DNA damage response players. Now, this process where a lot of proteins sort of pile on top of each other, create these big nuclear foci spanning the sites of double-strand breaks, this process is highly structured and very precise and streamlined. For example, if there is a difference of 2 seconds between the recruitment of protein A and protein B, this difference will be maintained, and protein A will always be the first, and 2 seconds later, protein B will come in. And there are a lot of checks and balances. So this actually is a finely tuned process, and a lot of work in this regard has been done recently in the lab of Dan DeRoche in Toronto, and I will mention that later on. So this is fine-tuning actually is the name of the game in this enormous network. This also set the scene for the activation of the so-called transducers, which are simply protein kinases that can phosphorylate a multitude of effectors. The transducers can feed back to the sensors, thereby amplifying the DNA damage signal, which is sort of irradiating from the site of the double-strand break. And then the effectors, of course, are involved in a variety of processes, and all of which finally ends up either in cellular survival following successful repair and modulation of numerous signaling pathways, or can result in cell death. And of course, the delicate balance between these processes with the final outcome, either cell death or cell survival, is a very active area of research. One thing we realized recently is that as opposed to our old view of, which was like the cell has a way to estimate the extent, the amount of damage, and then chooses either to activate programmed cell death or the cell survival mechanisms. We now know that both of them are activated at the same time, and this delicate balance actually takes place at the time when opposing mechanisms are active. And again, a lot has to do with gene expression. So with so many proteins involved in maintenance of genome stability, no wonder that we have these genomic instability syndromes that are typically characterized by sensitivity to genotoxic stresses, tissue degeneration, cancer predisposition, like AT. But again, the special point I want to make about this is that we always thought that milder mutations or heterozygosity for the severe mutations can contribute to chronic conditions. And here are a few examples. And we're particularly interested in premature aging. And so we predicted or hypothesized that the load on public health due to sequence variations in genes that encode proteins that are involved in maintenance of genome stability, actually that load is much higher than these very severe but fortunately rare genomic instability syndrome. And in fact, scattered evidence in the literature is now emerging, and we have with a lot of work which is being done now with mouse models that shows that indeed this is the case. For example, and I want to get into the data for lack of time, I'll just mention very briefly that we found that there is synthetic lethality between ATM and the Werner syndrome protein. This is the famous premature aging syndrome. But more interesting than that is the fact that mice that are double heterozygotes in the ATM and the Werner loci, so they are heterozygous for a null mutation in the ATM locus, and they are heterozygous for a null mutation in the Werner locus, also have a phenotype. And this genotype is interesting because this is a real genotype that exists in human population. For example, in this country, the estimation is that between 10,000 and 12,000 people have this genotype. Each one of us is a carrier of few scores of such mutation. And these mice actually start dying at the age of between 7 and 12 months, and we suspect that they have metabolic syndrome. They show tumors, premature aging, and radiosensitivity. And again, this is a genotype that exists in human population, and so 10,000 individuals is probably it's probably not a huge fraction of the population, but it is a huge number of people, of individuals. So actually, this old slide that used to be a hypothesis slide is now coming true. But of course, these disorders are the ones that flag the genes, and most of these genes were cloned because of people's interest in these phenotypes, and AT is no exception. And the ATM gene, ATM stands for AT mutated, was identified in our lab a while ago, and in what was a very typical race to a gene via positional cloning, very typical to that time when the genome technology was just emerging. And then we switched gears to understand what the ATM protein does. And so I'll show you the bottom line of what we think these days, and of course, this is a result of work in many laboratories, including ours. This protein is indeed missing or completely inactivated in AT patients. It has a predominant nuclear fraction, but it also has a cytoplasmic fraction, which is sometimes hard to detect, but it is important. It's a large protein, and it has this interesting PI3 kinase motif in the carboxy terminus, which we identified right away as we were sequencing that cDNA, of course, starting with the 3' end. But in fact, our lab, and independently the lab of Mike Castan, 3 years later, identified its or actually nailed down its activity as that of a protein kinase, specifically serine threonine kinase. And we also found its first documented substrate, no wonder P53. And we noticed that its activity is enhanced following the induction of double-strand breaks, what we call ATM activation. And it phosphorylates preferentially serine threonines followed by glutamine. It's preference, but in fact, it can phosphorylate serine and threonines followed by other amino acids. For example, one of the autophosphorylation sites that we found on ATM itself is an SD. And it is indeed the primary transducer of the double-strand break response by phosphorylating numerous, and when I say numerous, I really mean numerous, multitude of DNA damage response players. And it's a multi-layered phosphorylation network because it also phosphorylates other protein kinases. Check 1 and check 2 are good examples, but there are others, and they in turn phosphorylate their own targets. So many proteins can be phosphorylated on many sites in an ATM-dependent manner. Some of these sites would be ATM-mediated, and the others will be ATM-dependent because they are phosphorylated by other kinases, which are themselves activated by ATM. And structure for ATM, not even the kinase domain, is not, at least not published yet, and this is actually a major stumbling block in understanding how ATM is activated. So ATM is definitely a transducer. We found early on when we didn't, I think, appreciate enough our own finding that a good portion of ATM is actually recruited to the sites of the damage, like the other proteins, and many of the ATM-mediated phosphorylations actually take place within that protein conglomerate. The rest stays presumably in the nucleoplasm. This is actually a subject of an ongoing, I would say, discussion, debate, controversy in the field, how ATM is activated, what is the actual physical trigger. It's unclear yet, and I don't have time to get into all this. I'll just mention that ATM comes out of this process decorated with many post-translational modifications, many of which are autophosphorylations. And again, just a few comments. As opposed to our old view of ATM being inactive or silent or dormant in untreated cells and then explosively active in cells following the induction of double-strand breaks, we now know that there is actually basal ATM activity going on, serving the ongoing ATM duties, which I will explain later, and then it's modulated according to the need. So it's a very sophisticated regulation mechanism, and of course, the activity is markedly enhanced following the induction of double-strand breaks, and there are different modes of activation. For example, this one, which was recently published from Tanya Paul's lab, where ATM is activated directly by reactive oxygen species, and this mechanism is different completely from the mechanism that follows the induction of double-strand breaks, and we suspect that there are different spectra of substrates following different modes of activation. So still relating to this ongoing discussion about the importance of these autophosphorylations and the other modifications, we do think that these post-translational modifications make a difference. Of course, they are part of ATM activation. And I also should mention that ATM, as you know, is a member of this distinguished family of most of these proteins are protein kinases. They're quite large protein. They all share these motifs. And I'm now highlighting the DNA, the catalytic subunit of the DNA-dependent protein kinase, which is best known for its involvement in the non-homologous end-joining pathway of double-strand break repair, and ATR, which is best known for its being responsible for the response to start replication forks. But the reason I'm highlighting all three of them together is that we're learning, and we've learned this over the years, and we're getting quite solid evidence that these three proteins collaborate and are partly redundant. And when ATM is not there, actually, at least partly, ATR and DNA-PK can sort of compensate a little bit for this loss. And that's why in cells from AT patients who usually do not have ATM at all, we do see all of these responses, and they are carried out to various extents by DNA-PK and ATR. So DNA-PK actually does a lot more than just being a cornerstone of the non-homologous end-joining of double-strand break repair. And there are very interesting intimate relationships between these three proteins. And this redundancy actually is one of the reasons that we hope we'll be able perhaps to identify drug therapy for AT patients, and the mechanism would be enhancing these two guys, or at least one of them, to work harder and compensate better for the loss of ATM. We know that even partial compensation of, say, 15 to 20% of the normal activity would make a huge difference for these patients. So, and down here are the effectors, basically ATM substrates. So this is the map of the well-documented substrates, on each of which there is at least one paper. But as you know, phosphoproteomic screens carried out here by Steve Elledge in our lab and other labs identified numerous other putative ATM substrates. And if we feed all of this list into the algorithm that actually draws for us these nice diagrams, we get this kind of picture. There are about 1,100 proteins here. And you may think that ATM is actually a promiscuous kinase. We think, due to many reasons, that ATM is not promiscuous kinase, and most of these, or even all of these phosphorylations are important. Some may be more important than the others, but in the end, and again, fine-tuning is the name of the game, you need all of the musicians in this orchestra to be on the stage in order to get the perfect sound. And here is one example, going back to P53. So early on, we found this direct phosphorylation of P53 on serine 15 by ATM. We quickly realized that this phosphorylation alone could not account for the stabilization and activation of P53 following the induction of DNA damage. And then we turned our attention to MDM2, which at that time was the only ubiquitin ligase of P53 known, and identified this phosphorylation here, which is still, just this phosphorylation is still very active area of research. Just yesterday, a new paper came out on this phosphorylation. And then we looked at the degradation of MDMX, which is a strong inhibitor of P53. Basically, this is the summary of the subnetwork in the DNA damage response that is responsible for a very simple, specific aim to activate and stabilize P53. And all of it is actually, all of its branches are governed by ATM, are controlled by ATM. So ATM has a very strong grip of this pathway on one hand, and on the other hand, it can fine-tune it by phosphorylating, by working hard and phosphorylating so many different proteins around P53. So you could think of a dozen or 2 dozen other pathways like this, and you quickly come to number of substrates, which is in the hundreds. So when you look at these 1,100 substrates, this is their functional division, and of course, you can see here almost every area and every arena of cellular metabolism. Now, so with all of these numerous amount of players in the DNA damage response, are all these proteins actually dedicated to the DNA damage response. So much of the human genome is actually devoted to maintenance its own stability. Of course not. There is a core of a small core of proteins which are indeed bona fide DNA damage response protein dedicated to this response. What about all the others? All the others actually are busy doing other things when double-strand breaks are not induced. But when the need arises, they are called to action. They are actually recruited physically, as you can see, by physical recruitment to the damage site, and functionally to serve, at least for a few hours, the DNA damage response. And we're now beginning to think of ATM in the same terms. It does other things, but then is recruited and is activated in order to activate this entire system following the induction of this critical double-strand break. So this is actually a very simple and very economical and ingenious principle. And here is one very typical example, one of many. This is work that we did a few years ago. This is histone H2B monoubiquitination, which is carried out by an interesting functional module. And the heart of it is the ubiquitin ligase, which is a heterodimer of these two ring finger proteins, RNF20 and RNF40. And Lilach Moyal in the lab found that this whole functional module is actually recruited physically to the site of the break, phosphorylated on several sites. She mapped them all. And basically, this process is going on all the time, hand in hand with transcription. There's huge literature about the role of histone H2B monoubiquitination in transcription. And one of the things that happened is that actually this ubiquitination by itself helps to relax the basic 30 nanometer fiber of chromatin, and this is exactly what happens here. Basically, this functional module does here the same what it does in the transcription context. It monoubiquitinates histone H2B in the area of the break, and this is actually critical for a very late part of the early stage of the DNA damage response, the recruitment of the actual repair proteins. So if we abrogate this pathway, and there are many ways to abrogate it, we simply abrogate double-strand break repair, and this leads to cellular hypersensitivity to ionizing radiation and so forth. So this is a very, very typical example of this principle. And we're now continuing to study, actually we're comparing notes with the transcription people, and we want to see the similarities and differences between the way this complex behaves in this context and in the context of transcription. So, like I said, a lot of protein post-translational modification, typical signaling, and this is mediated by protein phosphorylation and protein modification by the ubiquitin family proteins, primarily ubiquitin, SUMO, and NAD8. And this actually spreads over megabases of DNA. Primarily, the main vehicle is the phosphorylation of histone H2AX. And here are the two of the main players in protein ubiquitylation. This is a slide I borrowed from Dan de Roche that shows you in greater detail some of the players in K63 and K48 mediated ubiquitylation at the damage site, and I don't have time to take you through this slide in detail. Here's the checks and balances and a few other players that are not shown in this slide. But what I wanted to draw your attention to is these question marks. And so on one hand, there's a lot of phosphorylation and a lot of protein ubiquitylation going on at the damage site. We know a lot about the substrates of phosphorylation. Phosphorylation sites are mapped. The functional significance of this phosphorylation is known. At the same time, so much ubiquitylation is going on. And I'll show you in a minute how we'll detect it. And yet, the only definite substrate is histone H2A, the well-documented substrate, and its derivatives. And the others are in question mark in this review slide. So the question is, what are the other, probably numerous, substrate of ubiquitylation, SUMOylation, and NADylation at the damage site? And this is, of course, an active area of research and one major project in the lab. And just to show you how further evidence about the importance of protein ubiquitylation at the damage site. So you're all aware of the different type of linkages of the ubiquitin chain, and of course, the K63 is primarily for modification of protein function, while the others, primarily K48, leads to protein degradation, usually via the proteasome system. And this is actually a very productive collaboration that we have with Christian Ramadan and his student Judith Eller at the Gray Institute in Oxford. And this is how we see these ubiquitinations using antibodies specific for K63 chain and K48. And you can see actually, this is actually induction of localized damage using focused laser beam. So if this is the entire nucleus, actually the damage is induced in a small defined sector of the nucleus. This actually helps to sharpen, to make the readouts much clearer, and this is a tool that all of us use quite frequently. And you can see here the time course. This is K63, and this is K48 that goes up very, very rapidly and then goes down, as you can see here. And in fact, the extensive K48 mediated chains, which are associated with, or presumably associated also at the damage site with protein degradation, sort of crosstalks with a previous finding in our lab about massive accumulation of proteasome particles at the sites of the damage. So this is just treatment of cells with a radiomimetic drug, and you can see here, this is actually a subunit of the core proteasome. And you can see here the accumulation that coincides with gamma H2AX. And here, again, using the laser lines, you can see that there's actually a massive recruitment. And we know that it's not just this protein. It's a whole proteasome, and I'm skipping a lot of data here. And this is actually dependent on ATM and also dependent on PA28 gamma, which is quite an interesting new substrate of ATM. As you know, you're all familiar with the basic structure of the proteasome, and this is the 19S lead. There's also an alternative type of lead, the 11S lead, which is a heptamer of the PA28 subunits. There are alpha, beta, and gamma is the nuclear one. So this is a special type of proteasome, and Adva Levi-Barda in the lab found that it is indeed a DNA damage response player. So typically, if you knock it down, it abrogates double-strand break repair. It impairs the important critical balance between the two major repair pathways. This confers radiation sensitivity, and it is also an ATM substrate. So again, we see that there is actually, we see all the components of the ubiquitin proteasome system being recruited massively within minutes to the damage site, clearly indicating that something is going on here, and proteins must be degraded quickly at the sites of the damage. And so we're actively pursuing them. And there's one new piece of information here, which is quite interesting. The knockout mice for P28 gamma have very mild, they were made by Lance Barton, and their phenotype is very mild immune deficiency. Or in other words, basically almost no phenotype. But when we irradiate these mice, which are otherwise perfectly healthy, normal lifespan and everything, they're tremendously radiation sensitive. So they do have a phenotype which is revealed following their radiation, again attesting to the importance of this little protein in the DNA damage response. I'd like to switch now to, for a few minutes to this branch of the system, modulation of gene expression. We've been doing a lot of work on the transcriptome dynamics. Of course, previously we used chips, and recently we switched to deep sequencing, RNA-seq and chip-seq. And we're quite convinced that there is a deep, extensive, important, slow layer of the DNA damage response at the gene expression level. So, and this is work which is about to be submitted. And this is the team in our lab that carried out the work, and this was a fantastic collaboration with the group of Mary-Laurie Aspo at the Max Planck Institute in Berlin. And basically, this kind of analysis yields terabytes of data, and I won't bother you with the statistics and even gene lists, just give you the bottom line. So the functional enrichment is, and by the way, these are p-values, so these are not numbers. So this is a very rigorous statistical analysis done by Rani Elkon in the lab. You can see here the major class is cell death, which is quite interesting. This is the upregulated genes. Now, the repressed genes, they're all in the arena of cell cycle progression, nuclear division, DNA synthesis, which again tells us that in addition to those nice and elegant signaling mechanisms that take place at the protein level with phosphorylations, ubiquitylation, proteins going up and down, the maintenance of sustained cell cycle checkpoints is actually also supported by this change in the transcriptome. We were amazed to see this proportion. We knew all along that p53 was the major player. But in fact, using deep sequencing, 95% of the response was p53 dependent, and the rest was NF-kappa B dependent. And the major class among the p53 induced genes was, again, apoptosis. We have a lot of new insights also about p53 mediated regulation of gene expression, but it's, I don't have time to discuss this. But I'll just want to go back for a second to this slide, basically showing you why, now we understand why ATM takes such good care of controlling very, very meticulously, in a fine-tuned manner, the level of p53. It's one of ATM's major arms in the DNA damage response with regard to gene expression. And of course, the NF-kappa B activation is also governed by ATM. Most of the work was done in the Miyamoto lab in Wisconsin. And so p53 and NF-kappa B dependence means ATM dependence. And I'd like to show you just another example. And in fact, we've already heard this morning about the histone methylations that are involved in gene activation. So here is again H3K4 methylation. And this is actually an average over several hundreds of, the promoter of several hundreds of genes which were activated. And as you can see here, you can see the elevation in this post-translational modification. This is the transcription start site. It's interesting that it has this bimodal distribution on flanking the transcription start site. And all of this is ATM dependent. When we add the ATM inhibitor, all of this disappears. And of course, there is a very nice correlation between this histone modification and the expression level. And the same is true also for monomethylated K4. And again, you see the bimodal distribution, which is most evident following the induction of DNA damage. And all of this disappears. It's erased by the ATM inhibitor, again showing you the absolute control of ATM on this process. So in fact, the title of my talk is identical to the title of a review that we published a few months ago. And so what is this N more that actually occupies about 1/3 of the review and summarized in this cartoon? So these are the pathways in which ATM involved outside of the DNA damage response arena. And you can quickly see, again, I won't get into the details. It's all in the review, but you can see that redox balance is very important. Mitochondrial homeostasis is very important. A lot of work in this regard is being done now in Mike Kasten's lab. And so it seems that ATM is actually involved in various aspects of cellular metabolism. And in that regard, I'll mention briefly the work done by Hadar Sharfi, a grad student, officially in our medical school, but in fact she's a joint student of our lab and lab of Michael Yaffe, our host. And in fact, the work was done entirely in Mike's lab. And basically, what Hadar did was a metabolomic screen, a typical metabolomic screen, following the induction of DNA damage, specifically by ionizing radiation. This was done in primary human fibroblasts. That's important. And what she noticed was a marked metabolic shift involving major metabolic pathways. And here are a few examples. And this is really quite impressive. And of course, there is strong ATM dependence. And she was also able to identify some ATM substrate in major metabolic pathways. And the metabolic shift actually is quite interesting because it's noticed it's quite longer compared to the time course that we're used to work with when we look at the double-strand break response or double-strand break repair. And so this is actually quite impressive. In fact, even the addition of the ATM inhibitor alone without radiation to the cells caused quite a marked metabolic shift. So we have to bear in mind, having said all this, that what we do in the lab by irradiating cells or treating them with radiomimetic drug, this is our favorite one, neocrosinostatin, is different from what's happening in the tissues of our body, as I mentioned before. And in fact, all this morbidity and mortality that is caused by defects in the DNA damage response is caused because of incomplete or suboptimal capacity to deal with this kind of damage, the everyday damage. And here I'm going back to this mild hypersensitivity of ATM to the other DNA damaging agents. So in fact, taking genome stability or talking about genome stability in a wider sense, of course, it's the DNA damage response to numerous DNA damaging agents, DNA replication, DNA transcription, which involves the occasional formation of single-strand breaks, nucleotide metabolism, and the resolution of such structures, which are occasionally caused in the course of normal DNA transactions. In fact, we now have evidence that ATM is behind all of this. And in fact, what it does, it phosphorylates various enzymes in all of these aspects of cellular metabolism, thereby enhancing their activity. I'm not getting into the details, but basically we think that the ongoing everyday duties of ATM in a tissue cell is actually to cope with these lesions by optimizing the cellular response to genotoxic stress. So we're still in the nucleus, in the DNA damage response arena, but we're taking ATM beyond the mere double-strand break response, and we think that this is actually based on this basal kinase activity, which of course is tremendously enhanced if we induce double-strand breaks. And finally, how can we use all this to go back to the patient bedside? We're carrying out a high-throughput drug search. I don't have time to go into the details, so I'll just mention our collaborators in this regard. And in fact, we already completed a screen of 300,000 drugs, came up with one hit. But this one hit actually seems to be doing what we were hoping that will happen. So at least we have proof of principle that this kind of screen can be carried out. And actually, they will probably teach us or give us some more insight into the DNA damage response. Finally, 2 credit slides. First, funding. And we really have to thank all the funding agencies that fund us in these trying times for science funding. And some of them are in Israel, some are in this country. And finally, when we PIs say we in such talks, we usually mean somebody else. So who are we? This is our group. I think I mentioned names of a few of these people. And everything that I've told you wouldn't have been done without this fantastic group of extremely talented and devoted students. Most of them are students. Thank you for listening.